Okay, so we're going to pick up where we left off before. We were sitting at the beginning of meiosis 2. These are cells that are essentially sitting in the beginning part of the process, which is called prophase. And the way I have them drawn, I also have them lined up in the middle of the cell. So this is more or less what it's going to look like when the cell is sitting in metaphase as well. So I'm going to label this cell as both prophase and metaphase. It's kind of a snapshot of what both of them might look like. And then therefore this cell down here is sitting in the same two stages, prophase 2, and then they line up in the middle called metaphase. And again, we're in the second half, that's metaphase 2. Now the next thing is these chromosomes are going to have those spindle fibers form again down the equator, pulled apart. So there's my little centrioles and my spindle fibers. Centrioles, I'm going to draw them fast. Spindle fibers. These chromosomes are going to get split down the middle here and pulled in opposite directions. So if all goes well, the next cell would look something like a little pink one and a little pink one getting pulled to opposite sides. Long pink one and a long pink one getting pulled to opposite sides. Long green one, short green one, short green one. And I'm trying to draw them as if they were moving, so that's why I've shown them kind of crooked, bent. So again, it's like the spindle fiber has grabbed hold of them and is pulling them over here, pulling them away from the equator of the cell. Okay. Pulling them away from the equator. Now, away from the equator means that we're actually sitting in the phase known as anaphase again. And this is anaphase 2. Before we go to the last step, I just want to emphasize what has now gone on. These are called sister chromatids. They are exact copies of one another. We copied these all the way back here when we first made a duplicate, comparing these two cells to one another. So these are called sister chromatids. Earlier, the homologs became separated from one another when we pulled them here and here. But now, we're separating our sister chromatids. Sisters got separated. Sisters got separated. Sisters got separated. An easy way to remember this is the phrase, sisters separate second, meaning in the second half of meiosis, our sister chromatids are separating. Okay, let's draw our final result. Assuming this cell pinches inward in the middle, just like we saw earlier, we call that cytokinesis. We're going to end up with this little cluster right here becoming one cell. This little cluster over here becoming a second cell. Let's draw one side here and the other side down below. And I'm going to do the same thing over here. Little guy, longer one, longer one. And again, that's after the cytokinesis pinching inward has formed. This pinching inward is referred to as cleavage. It's like the cell is being cut or cleaved like a meat cleaver. And that would mean that cytokinesis has to occur before we can get to our final cell here. So cytokinesis is happening on each of these little arrows. Okay, so we end up with the 4 sperm cells pictured here. These two are alike one another, and these two are alike one another, but this cluster up here is different than the cluster down here. And the reason for that all goes back to how I line them up in the middle back at this step. Now keep in mind, we also said that there might have been crossing over to occur. If that were to happen, then you're actually going to see a little variety amongst these two, meaning you're going to have a little variety here that was different than what I currently have drawn, and that same variety might become evident in the sperm. So here we are, 4 sperm cells that have been produced, probably only one, could potentially go on and fertilize an egg. So if we assume that this is the egg over here, eggs are enormous compared to sperm, so we're only looking at a small portion of an egg. The egg would be massive. I'm going to go ahead and draw in a few chromosomes that this egg would contribute to this developing potential embryo. They're going to have a green one from their own father, so I'm going to make it the little one. And then they're going to have one from their own mother that's longer. So let's just say those are the ones that ended up in the egg. And we can pick one of our sperm at random to join up and fertilize this egg. So if that were to happen, you end up with a little zygote. The zygote is the first cell of your potential embryo. And with the two that I've chosen, I'm going to end up with two long pink ones and two short green ones. Just so happen to be that way. What that tells me is this zygote, potential child, just inherited a chromosome from their grandfather on their dad's side of the family. And they also inherited a chromosome from their grandfather on their mom's side of the family. They've inherited chromosome number 12 from their grandmother on their dad's side of the family, and inherited chromosome 12 from their grandmother on their mom's side of the family. So this is how somebody might actually get some traits of their grandparents. You may end up knowing someone, or maybe even you, similar to grandparents' traits. Right? The chromosomes are passed through the generations. Now if we compare our ending sperm cell to the very first cell we started this process off with, over here, how does the chromosome number compare? In this one, we have two distinct chromosomes. Well, in pairs, so we actually have 4 total chromosomes. Down here, we only have 2 total chromosomes. We have half as many chromosomes in our gametes. That's an important takeaway from meiosis. I'm going to add a little note. We have half the number of chromosomes as the original cell. Meaning in humans, this is a cell that has 23 chromosomes. There's a name for this. Earlier we referred to the first cell as diploid. The cell that results at the end of meiosis is called haploid. And that definition is having half the number of chromosomes as the original cell. We only